Welcome back to our YouTube channel Histology. In this video we will discuss human body tissue fixation and preparation. From cell morphology to the diagnosis of various diseases and comprehension of different physiological processes, much research is done through tissue sample examinations in histology, pathology, and biomedical research. An integral part of these fields is tissue preparation and fixation. Proper fixation and preparation help preserve tissue structure as well as biological components, thus allowing pathologists and researchers to study them with precision under a microscope. In this paper, I will summarize the importance of tissue preparation, how human bodies seem to fixate in various ways, and what happens after tissue processing is done. Overview of tissue preparation to preserve biological tissues for study. Several steps have been undertaken in tissue preparation. Tissue specimens degrade rapidly through autolysis, or self-digestion, and putrefaction, or microbial breakdown, if not properly prepared. The tissue preparation procedure can be generally categorized into the following steps. 1. Specimen collection. 2. Fixation. 3. Dehydration. 4. Clearing. 5. Impregnation. 6. Sectioning. 7. Staining. Therefore, this step would be crucial to preserve the natural tissue architecture and cellular integrity, which is necessary for accurate microscopic examination. In other words, every step is critical. Point 1. Sample collection. The first step towards tissue preparation would be tissue samples derived from a human body. Tissue samples can be obtained through various methods such as biopsy, punch, needle, excisional, surgery, and autopsy, depending upon the clinical situation. In order not to undergo decay, tissues have to be obtained quickly and completely. It would be ideal to treat fresh tissue immediately so as not to allow morphological or chemical changes to take place. In some cases, tissues may be preserved if immediate processing is not feasible. Frozen as an interim measure, or in a fixative. These measures, however, are only palliative. Optimum fixation is necessary to preserve tissue in the long term. 2. Aggregation fixation, which prevents tissue decay and preserves tissue structure, is the most critical step in tissue processing. The major aims of fixation include preserving protein bonds, stabilizing cells and tissue structure, and halting the growth of bacteria and self-destruction. Why fix? Preserve morphology. Fixation ensures that the cells retain their inner structures, size, and shape so that a histological examination may be carried out with accuracy. Anti-putrefaction and anti-autolysis. Fixatives inhibit those enzymes which are capable of breaking down cells and check the proliferation of microbes. Improve stainability. Once tissues are properly fixed, stains are taken up perfectly and stains appear very vivid under the microscope. Fixatives types: chemical and physical. Fixatives can broadly be classified into two categories. Point 1. Chemical fixatives. The tissue structure is stabilized by the formation of crosslinks between proteins and other cellular constituents through chemical fixatives. The most common method of preserving tissue is through chemical fixing. These can be differentiated as coagulant as well as non-coagulant fixatives from this category. Aldehyde-based fixatives. The most commonly used aldehyde-based fixatives for human tissue are formaldehyde and glutaraldehyde. Formaldehyde. The most widely utilized fixative in clinical pathology is formalin, a 10% formaldehyde solution. Protecting tissue integrity by forming cross-linkages through reaction with proteins is the role of this fixative. Good overall morphological preservation and rapid penetration rate into tissues characterize formaldehyde. Although glutaraldehyde preserves finer cellular characteristics better than formaldehyde, it penetrates tissues more slowly and is therefore often used cautiously for electron microscopy or smaller tissue samples. Alcohol-based fixatives. Fixatives based on alcohol. The fixatives based on alcohol are methanol and ethanol. They are usually applied because they dehydrate the proteins and precipitate them and thus are used frequently in molecular biology researches when nucleic acids need to be conserved. Agents oxidizing. Another fixative employed mainly for electron microscopy is osmium tetroxide. Lipids and membrane structures are preserved, which is very important for ultrastructural research techniques. Frozen sectioning of tissues, or cryosectioning, is an important procedure for tissues that need to be studied right away. Freezing can be achieved either in liquid nitrogen or with some cryoprotectants. 3. Dehydration. After repairing the tissue, it should undergo dehydration to remove remaining water in the cells. Water in the tissue makes it hard to embed and section, hence dehydration is normally achieved through the escalation of the concentration of ethanol from 70% to 100%. The process of dehydration gradually avails the prevention of tissue damage that can be incurred due to sudden cutting off of water. 4. Clearing. After dehydration, it is important to eliminate the fluid content of the tissue and to replace the alcohol used for dehydration with a material that can dissolve both the embedding mediums and alcohol, generally paraffin. At this stage, many do like to use toluene or xylene as they are compatible with paraffin wax. Additionally, the process tends to increase the transparency of the tissue, making it easier for the embedding medium to penetrate the tissue. 5. Inclusion. The tissue is embedded in a medium that permits sectioning in the final tissue preparation step, embedding. Paraffin wax is the most common for routine histological study. Now alcohol-free and free of all contaminants, the hot paraffin wax flows into the tissue and solidifies as it cools. In this way, a tissue block can be formed that can be cut into very thin slices to be made into microscope slides. For electron microscopy, one other choice for an embedding medium is resins, such as araldite or epoxy, that offer much better resolution for ultrastructural studies. 6. Dividing. Tissue samples embedded in either paraffin or resin are sectioned into thin slices, around 4 to 5 micrometers for light microscopy, with the aid of a microtome, which is essentially an instrument that cuts tiny pieces of tissue from a block attached to the instrument. For electron microscopy, ultra-thin sections, measuring 50 to 100 nanometers in thickness, are cut using an ultramicrotome. These tissues are then attached to the glass slides for further processes like staining. After the above steps. 7. Staining. It is rather difficult to distinguish cell features in unstained tissue sections as they are nearly translucent. Contrast between various tissue structures is enhanced through staining. Depending on the tissue and structures of interest, several types of stains are used. Many commonly used stains include hematoxylin and eosin, HNE, the stain which histologists use the most. Hematoxylin stains cell nuclei blue-purple, and eosin gives tissue components, including the cytoplasm, a pink stain. This will highlight different tissue components clearly, which will be important to understand cell morphology. PAS staining is useful in diagnosing several disorders, such as infections caused by fungi and diseases related to the storage of carbohydrates such as glycogen. Mason's trichrome. These stains are used for the analysis of connective tissue so that one can differentiate between the muscle, collagen, and erythrocytes. It is commonly used to test for fibrosis in tissues. Immunohistochemistry, or IHC for short, is one of the most advanced staining techniques that can identify specific proteins in tissues through the aid of antibodies. This proves to be particularly useful in cancer diagnosis since IHC can identify tumor markers. Conclusion. Tissue fixation and preparation is part of the pathology and histology. The tissue is preserved by proper fixing tissue morphology. Further, it prevents deterioration and allows research and accurate diagnosis. There are many fixation procedures available, each offering unique advantages for the type of tissue and study to be undertaken, and ranging from chemical fixatives such as formaldehyde through to physical methods like freezing. Following fixation, there is a further set of procedures to be undertaken: dehydration, cleaning, embedding, sectioning, and staining, all of which require individual care and attention to ensure that tissues can be inspected under the microscope without any issues arising from previous steps, excepting in fixatives. Fixatives like picric acid and mercuric chloride cause protein coagulation and precipitation, which confers the impression of a three-dimensional network of proteins that maintains the shape of tissues. Due to their toxicity, these fixatives are rarely used today. 2. Body fixatives, freezing, where chemical fixation is not liked. The examples include studies on enzyme activity or in certain immunohistochemistry, fast freezing of tissues is mostly applied.